Okay, today we are looking at cell division in terms of meiosis. By the end of this lesson, you're going to be looking at how to draw and describe the steps involved in gametic cell replication, in other words, meiosis. Okay, let's look at gametes. What are gametes? Remember, there are two types of body cells in your body. The first one is called gametic cells, in other words, eggs and sperm, or if you're a plant, it's pollen and the ovum. The other type of cell is called a somatic cell, and these are every other cell in your body except for the egg and sperm, the gametes. Okay, what are gametes? Because sex cells, they are haploid, which means they have only one set of chromosomes. In humans, they have 23 chromosomes and they are haploid with 23 chromosomes. When the sperm from the male and the egg from the female fuse together during conception, this is called fertilization. This is where the nucleus of the sperm enters the nucleus of the egg. They combine and fuse together to make one diploid cell, and that one diploid cell is called the zygote. It's now a diploid cell because the 23 chromosomes from the sperm fuse with the 23 chromosomes of the egg to produce a diploid 46 chromosome cell. Okay, that diploid cell, the zygote, then goes on to divide mitotically using mitosis to grow into an embryo, into a baby, into a child, teenager, and adult. Cell division. Here we've seen this diagram before. We're looking at meiosis now. Okay, meiosis. What's the use of meiosis? What's the purpose of meiosis? The purpose of meiosis is to produce gametes or sex cells, eggs and sperm. Where does it occur? Well, it occurs in the ovaries of the female and the testicles of the male, or the anthers of a plant. Okay. What type of cells does it produce? Remember, with mitosis, the cells that are produced are genetically identical to the parent cell. Well, with meiosis, the cells are genetically different or dissimilar to the parent cell, and they're also dissimilar to each other. Why meiosis? Why do we possibly need to have another type of cell replication? Why can't we just have mitosis where all the cells are exactly the same genetically? Well, remember, we are part of a larger population. Okay, the most important reason that meiosis has evolved is to produce genetic variation for a whole population. If a population has exactly the same genes with no genetic variation at all, then they may all succumb to the same diseases or environmental factors. Let's say there's a disease that came along and everyone in your population in the town or the village that you live in are exactly the same. That means they look exactly the same, they sound exactly the same, and they're all exactly this. They are all exactly the same, susceptible to the same diseases in the same way. Then it's possible that a disease may come along and wipe them all out, kill them all, every individual in the population. But if a population undergoes meiosis, then genetic variation within a population will increase. Then if a disease strikes, then not all individuals will die of that disease because all their genes will be slightly different and they'll all respond to that disease or environmental factor in a slightly different way. Meiosis results in 4 new cells that are genetically different from the original cell. This means that all eggs and all sperm are genetically different from each other, and that's really important, and that's why you look slightly similar but also not the same as your parents, and also you look slightly different to all of your siblings and the rest of your family. Okay, of course the exception there is if you have an identical multiple birth like twins or triplets. Okay, so meiosis is a 2-cycle process. We have meiosis 1 and then we go on to meiosis 2. Okay, so the very first cell we're looking at here is a diploid cell. Okay, we can see that it's still in the interphase stage. The chromosomes or the DNA is still in that chromatin phase. They are starting to condense, to pack together more solidly. But those chromatids are also replicating. So once they become chromosomes, they condense, and then the chromatids are going to start to replicate and go from a single chromatid chromosome to a double chromatid chromosome. They're going to be double-stranded. So this original cell here, can't see my cursor, the original cell is diploid, which means that they have 2N, and N is the number of sets of chromosomes. So they have 2 sets of chromosomes in the original parent cell. And you can see, where's my cursor? You can see that, there it is, you can see that the centrioles are starting to form at the top of that cell. The nucleus, the nuclear membrane is beginning to break down. There's some nuclear pores there, and these spindle fibers are starting to come out of those centrioles as well. They're going to migrate themselves around to each of the poles, one there and one over there. Okay, the second step we have here is meiosis 1. So all of these processes is in meiosis 1. We have a prophase, a metaphase, anaphase, and telophase, just like we did in mitosis, and we also have cytokinesis. Okay, so meiosis 1, if we look at prophase, we can see prophase 1, this is a really important phase in the process of meiosis, where we have the homologous pairs of the individual lining up. You can see that the nuclear membrane is starting to break down, and those spindle fibers are starting to expand and extend throughout the whole cell as well. Okay, at this stage here, we have those homologous chromosomes lining up, and you can see that in particular, this cell has only 2 sets of homologous chromosomes. Now, in a human diploid cell, we have 23 sets of homologous chromosomes, or in other words, 46 chromosomes. But you can start to see these chromatids are starting to overlap here a little bit. We have a dark blue chromosome or chromatid overlapping with a purple chromatid. And this place where they cross over is called a synapse or a chiasma. And this is a step called, which is really important for what we call crossing over or recombination of genes. Now, if we look down here at this diagram here, you can also see the homologous chromosomes here. Okay, and we've got some genes labelled A, B, and C, and I've just labelled this one as from the mother, and this one has been from the father. So these are homologous chromosomes. The mother has a genetically dominant gene A, dominant gene B, and dominant gene C, and the father has a recessive gene A, a recessive gene B, and a recessive gene C. So at this stage of prophase 1, we have some crossing over of those chromosomes happening. Okay, we can see that the mother's chromosome is crossing over with the father's chromosome, chromatid, and in the end, they cross those 2 genes, gene C, across from each other. So in the end, the mother's chromosome will have a dominant gene A, a dominant gene B, and then a recessive gene C, and then vice versa for the father. Okay, this is really important, which will increase the genetic variation overall of the 4 cells that will be produced. Whether these 4 cells are eggs or 4 sperm cells, they'll all have an increased variation of genes, different from that mother or parent cell. That one there. Okay, so also you can see some crossing over of these 2 homologous chromosomes. The same process is happening where those genes are recombining and just basically shaving off and swapping over from chromosome to chromosome. Okay, so this is a really important step. Prophase 1 is where that recombination of genes occurs. Okay, at this stage, we still have a diploid set of chromosomes in that cell. Okay, let's look at metaphase 2. Can't see my cursor. Here we have metaphase 2. The nuclear membrane is completely broken down. The spindle fibers are starting to attach to the centromeres of each chromosome. Okay, and you can see that the the homologous chromosomes have completely lined up in pairs at the equator of the cell or the middle of the cell. Remember, meta means the middle. Now, this word tetrad, lining up on the metaphase plate, means tetrad means 4. And of course, we've got 1, 2, 3, 4 chromatids. And that's what that means, tetrad. Okay. In anaphase 1, you can see that those double sister chromatids have been dragged away from each other. So we have the maternal homologous chromosome being moved over to this side and the paternal homologous chromosome being moved over to this side. Okay. The homologous pairs separate in anaphase 1, and then in telophase, that one cell starts to cleave off into 2 cells. Okay, the nuclear membrane starts to reappear, and then we have a full separation of those cells in cytokinesis 1. Okay, so this is all meiosis stage 1. And then we move on to a completely new phase of meiosis 2, where we've got our cytokinesis, and then we're going on to meiosis 2. So it starts off with prophase 2, where we have these haploid daughter cells now undergoing one more cycle of division in order to separate those sister chromatids. Okay, remember each of those chromosomes still has 2 sister chromatids on them. Okay, and we've got the genetically recombinant chromosomes, which are increasing genetic variation. This goes on, each of those 2 cells go on to metaphase 2, where our double sister chromatids line up, and each of those spindle fibers is attaching to the centromere of that chromosome. And in each of those cells, we are now having that splitting apart of those double sister chromatids into single chromatids. Okay, if we take this one cell, we can see that that's now starting to cleave off, and the nuclear membrane has formed around in telophase 2. Okay, and is now producing those 4 genetically different gametes, eggs or sperm cells. Okay, so remember, we've started off with a diploid cell, one diploid cell. We end up having 4 haploid cells. So we're going from a 2N cell to 4 1N cells, haploid cells. Okay. All right, a little bit more information on how these cells are able to provide genetic variability. Okay, we have a law called the law of independent assortment. And just like up in this stage here, where we had those homologous chromosomes lining up and then recombining, we have these homologous pairs aligning here. So this is just another example. So you can see down here, there's a little key, a colored key. It says each gamete can be a different combination of maternal, which is red, the red chromosomes, and paternal blue chromosomes, depending on the orientation of the homologous pairs in metaphase 1. So in metaphase 1, where they're lining up here at the center of the cell, ready to be taken to their opposite poles, the maternal chromosome could be on either side of those of the middle. Okay, it doesn't matter which side. So you don't have to have all of the maternal chromosomes on one side and all of the paternal chromosomes on the other side. They can mix and match. It doesn't matter which side they go to. Okay, so you'll end up having maternal chromosomes on this side and then paternal chromosomes on this side. So it's just another source of genetic variation. Okay. All right, sex determination. We looked at autosomes. Remember, autosomes are the chromosomes in your body which provide the genes for normal cell function. Our sex chromosomes are our last pair of chromosomes, which is the 23rd pair of chromosomes. Now, they're going to be a pair of either X and X chromosomes if you're a female, or X and Y chromosomes if you're a male. Okay. So remember that humans have 23 pairs of chromosomes. The chromosomes that carry our sex genes are on both the X and the Y chromosomes. However, if you are a female However, their haploid egg cells will only carry one X chromosome to combine with the sperm during fertilization. So is it the mother or the father which determines the gender of the baby? The mother can only provide an X, so it can't be the mother's choice. The mother can only provide one type of chromosome. It's actually the father. The father can provide either the X or the Y, okay? So this baby that's going to be produced will be either XX for a female or XY for a male. Okay, our overview. Remember, sex cells are eggs and sperm in animals, or pollen and ovum in plants. Sex cells are haploid. They have one set of chromosomes, of 23 chromosomes. Meiosis results in 4 daughter cells, which are genetically different to the parent cell and also genetically different to each other. Meiosis provides much needed genetic variation within a population, and homologous chromosomes cross over or recombine in prophase 1. Meiosis is a 2-cycle process of sex cell division, and I've listed prophase 1, metaphase 1, anaphase 1, and telophase 1. This is all meiosis 1, and then we have cytokinesis happening in here, okay, in between these 2 steps, and then we go on to meiosis 2. Okay, guys, if you've finished taking notes, I want you to go to complete your meiosis and mitosis worksheet, the comparison worksheet, and I want you to complete the meiosis side of that. If you've finished that worksheet, then go onto this fantastic website and have a play around with all the different phases of meiosis. Last, we have a very clear diagram without me talking over it. You can pause the audio, pause the video, and just have a look at the steps.